Hello and welcome back to the next section of our Discovery Forum event. I'm Rich Scott, I'm the Chief Medical Officer at Genomics England, and this session is going to be on research, and we're going to give you three sort of little demonstrations of some of the research that's been going on recently with internal teams involved, but also with partners. And for those of you who want to join the bioinformatics track that is happening in parallel with this one and you're online, there's a link that you can click on in the chat that will take you there, and then the sessions will recombine for later in the day. So for those of you who are staying, we have three sections. We're going to start talking about Covid-19. We're then going to talk about cancer, some of the work that's been going on with the pan-cancer analysis, and finally we'll be joined by Matt Hurls to talk about some work in rare disease on hypermutation phenotypes. But to kick off, we're going to talk about a Covid-19 study, and I'm joined here with me in the room by Thanos Kustatanis, who is a genomic data scientist at Genomics England, and we're also joined from Edinburgh by Kenny Bailey, who is the PI of the Genomics study, with whom we've been working in partnership on the Covid-19 program over this last 18 months. And we're going to talk to you a little bit about the program and some of the recent findings. A lot of you will have heard about the program, and we've just got a slide to start us off, just to give you, to frame the program overall, and to give you an idea for the sorts of numbers and data sets that are involved. So this has been a partnership between us, Genomics England, and the University of Edinburgh, Kenny's team, principally in genomic, and actually many across the UK life sciences ecosystem. The programme has broadly been looking at the genomic determinants of Covid-19, and we've nearly completed the recruitment component of the programme with what will be approximately 15,000 people with severe Covid-19, largely admitted to intensive care units, and 15,000 people who are more mildly affected, non-hospitalised. And that's been an enormous effort in itself. Partnership for example, recruitment to the severe arm of the programme, supported across the NHS and with the NIHR Clinical Research Network, recruitment to the mild arm of the programme in partnership with the REACT research programme. And then it's about bringing that genome data together with other data sets that the research offer, the same research offer that is available to our participants in our other programmes allows, so linkage to longitudinal health data and so forth. And so now Kenny and Thanos are going to talk us through some of the most recent findings of the programme, and then we're going to have time, I hope, for some questions and answers. So firstly, Kenny, welcome. Thank you for joining us, and I think you're going to start and tell us a bit about the programme.
Thanks very much indeed, Richard. So if we go back a few slides to the timeline of discovery, as Richard said, we've been running the Genomics study. It stands for Genetics of Mortality in Critical Care for 5 years in the UK. I'm an intensive care doctor, and there's really a community of intensivists and research nurses across the whole UK who were already recruiting patients into this research study, fundamentally to understand the host genetic mechanisms of critical illness from a variety of phenotypes, mostly infectious.
I think while we work on Kenny's audio, what we'll do is we'll perhaps get Thanos to talk through some of the content in terms of the recent findings. Kenny was talking about the sort of infrastructure that was already there, that he and others had established from the point of view of the genomic programme itself. Also, a lot of what's been going on over the last year has been establishing and sort of working with partners across the UK ecosystem, as I mentioned. The study's benefited from Department of Health funding, from funding from LifeArc, from funding in kind from Illumina supporting sequencing, and also from the MRC and NIHR. One of the things that we've also done is adapt and update our infrastructure, so moving to a cloud research environment you've heard people talk about already, and the LifeBit platform. Kenny, you're back, and I think we'll be able to hear you. You look crystal clear.
Thanks, Richard, and it's a reflection of how closely we've been working together, Richard and Thanos and I, that we can speak each other's words. So yes, thanks very much, and apologies for the internet connection. We had at the beginning of the outbreak a major problem in identifying effective therapies. Of course, the recovery trial has done an incredible job of finding them. But at the beginning of the recovery trial, and I had the privilege of helping to set it up and run it, we had no idea what therapies were likely to be effective. And genetics has already helped us to do that. So initially, the first thing we learned about genetics came from a study from Spain and Italy, which is shown in this slide. Actually, the results were released in June 2020. And they had an incredibly strong hit with only a few hundred patients who would meet the entry criteria for our study here. They were already able to see this signal in chromosome 3, which we'll show again. It's still present in the data. And then in September 2020, we completed the first analysis of the genomic data. So a few months after the first critically ill patients appeared in the United Kingdom, we performed an analysis of 2,244 critically ill people. So that's the very extreme end of the susceptibility spectrum, where, as we now know and predicted at the time, most of the discovery power for genetics lies. And we discovered these 4 genetic associations, each of which has spun off its own field of research. And all 4 of them are potentially therapeutically targetable, although the 2 in chromosome 19, DPP9, and tyrosine kinase 2 had the most direct impact, and both of those influenced the choice of drugs that we are now testing in the recovery trial. Then in July of this year, we contributed to a paper that the genomic study contributes about 40% of the information that's present in this huge international collaboration, bringing together all the data from the many genetic studies, and found a few additional genes that are associated with severe disease and several that are associated with milder phenotypes. If we go to the next slide, and here I'll hand over to Thanos to tell you about the analysis. This is the latest analysis we've now conducted of over 7,000 critically ill people in the UK. This time, not using microarray genotyping as we used for all 3 of the previous studies that I talked about, but whole genome sequencing, which opens up a whole new range of opportunities for discovery, which we'll tell you about now. Thanos, over to you.
Thanks, Kenny. So, as Kenny said just a second ago, the thing is that all the previous studies had not sufficient sample sizes. So the first thing is to actually have a larger sample size of carefully selected cases, and also coupled with whole genome sequencing in order to assess all of the types of variation that could underlie COVID-19 critical illness. For this reason, we whole genome sequenced several samples of critically ill patients, and also we used as controls 100,000 Genomes Project participants, which were coupled with the same technology. And you can see here the breakdown of cases and controls that we used, and this spans across 4 different predicted ancestries, which is Europeans, South Asians, Africans, and East Asians. And by using this cohort and also a logistic mixed model regression, which can control for confounders, such as population stratification, that, for example, produce spurious signals, we performed a Genome-wide Association study, and you can see the results of this study here with this Manhattan plot, where the strength of association, the p-value is on the y-axis, and the position in the genome is on the x-axis. And what we annotate here is all of the signals that we found as statistically significant, and the related genes that we found that they are that these variants that we found having the statistical association having consequences in. For example, we also replicated the chromosome 3 signal, and we found many others, including on the HLA locus. And these signals, for example, here were found in the largest cohort that we had in Europeans, but we also coupled that with an analysis that combined the signals from all ancestries, and we discovered another 3 additional signals to these European signals, for a total of 25 signals, which brings the number of associations with COVID-19 to almost doubling of these novel findings. Then the other question is, when we see clusters of signals, such as here in a particular location, because they are closely together and linked through linkage equilibrium, the question is how we can distinguish signals that are independent. And this, we did it with fine mapping, and the very high resolution of the whole genome data that we had allowed us to distinguish signals that were like very proximally located in the genome, such as, for example, here on identifying 2 different chromosome 19 signals. And this is also because we don't have ascertained data. And in particular, for the HLA locus, which has a very complex LD, we also performed a bespoke analysis that we assessed different HLA alleles for the association with COVID-19, and we found the HLA DRB1 gene as being behind this association. And this has been also replicated by other studies. Finally, we also leveraged the summary statistics from the primary GWAS study to also check for associations with gene expression. And as you can see here, we also checked for colocalization of the GWAS signals with gene expression of several genes. And as you can see in this type of gene-based Manhattan plot, the colocalized genes with at particular tissues, for example, lung or blood, that we checked, and also the direction of change in expression, which is due to the particular risk-increasing allele. For example, in MUC1, the risk-increasing allele is increasing the allele frequency upwards. So this can also be the beginning of therapeutic interventions in terms of trying to fix these expression changes that are done by the risk-increasing variants. And I will hand over to Kenny to tell you a little bit more about the significance of all of these associations and signals.
Many thanks, Thanos. So I think it's fair to say that these are very fresh data, and we are still at the beginning of understanding their biological significance. And of course, unlike the previous set of 4 new discoveries that were reported in Nature last year, there are 23 robust replicated genes in the latest data, and numerous other genes that are implicated by GWAS and Mendelian randomization. So there is much that we have not yet explored about these new findings. They are, of course, in the public domain on medRxiv. So anyone in the audience who wants to can dig into them and draw their own conclusions. One of the striking signals highlighted in this slide is that there is a fairly clear separation between a group of genes that are well understood and implicated in host antiviral immunity. So of course, the interferon signaling pathway and the OAS genes have well understood roles in protecting ourselves against viral infection. And there are 5 genes with broadly concordant direction of effect. There's always some uncertainty in predicting direction of effect from genetics alone, but they're broadly concordant, suggesting that less interferon signaling is associated with more severe disease. One example, which is rather beautiful, is interferon alpha 10, where there's a single base change that leads to an amino acid substitution that probably disrupts the entire interferon molecule and may prevent it from being secreted at all. It might just be degraded. So it's predicted to be highly deleterious to the function of that gene. And in parallel, there's another single amino acid substitution in the last of the genes on that list, the LSCR1, which disrupts a part of the protein that is essential for nuclear import, which is part of the way that it conveys its signal, the interferon signal to the nucleus to affect gene transcription. So together, these leave us very certain that if you have impaired interferon signaling, you're more likely to become critically ill with COVID. And that's very interesting in the context of the results from the Solidarity trial last year, which treated patients with systemic interferon and found that there was no improvement in mortality. Bringing those two findings together, I think particularly now that we're now much more sure about the impact of the genetic variants, I think it's most likely that the timing and possibly the physiological location of the interferon is what's making the biggest difference. And it may well be that early in disease, you need interferon and antiviral immunity to control the viral load, and later in disease, host-directed immunity causing lung damage is the primary mechanism, and that's the category into which most of the other genes that we have found appear to fall. So a few examples are shown in this slide. There are several genes, BCL11A, TAC4. There are key factors for B and T cell development. CSF2 is the gene that encodes granulocyte macrophage. Colony stimulating factor, and is a key differentiation factor for myeloid cells, which are already implicated in the pathophysiology of COVID from autopsy studies and other data. And several genes with diverse predicted directions of effect that are implicated in idiopathic pulmonary fibrosis and other forms of lung disease, including MUC1, which is therapeutically targetable. MUC1 is a mucin, one of the primary mucin genes that creates the long glycopeptides that combine with water to form mucus. And there are existing agents that target that, and we have a fairly robust, in fact, a very convincing Mendelian randomization signal, which is a statistical tool to predict or to test evidence for causality, and that suggests that reducing MUC1 expression is likely to improve outcome. So there's much left to dig into, but I think we can leave the rest for questions if anyone's particularly interested in any specific findings. And if we go to the final slide, I'd be grateful to just take a moment, if you go to the next slide, please, to acknowledge the many people who've contributed to this work. The names of the primary authors of the paper that's currently under review are shown on this slide, and the group from the Genomic University of Edinburgh side and from Genomics England are shown at the top. But really the work that we've reported has been an absolutely colossal amount of work by about 1,500 research nurses in almost every single intensive care unit in the country. And it's entirely dependent on the generosity and altruism of patients who agreed to participate in this study at probably one of the most terrifying times in their lives. And so we're extremely grateful to all of them. We'll stop there and be delighted to take questions. Thanks.
Thank you, Kenny. I'm aware of the time. We had a little bit of a sort of technical hitch at the beginning, so I just wonder whether we'll sort of take one question before we moved on to the cancer focused part of this session. There's also a chance, I hope, for us to respond to people's questions in the chat if you have them. This has been a real collaboration between Genomic and many others and the Genomics England team. I guess one of the things that we sort of get asked quite a lot is, how are we using data from 100,000 genomes participants in this sort of work? Is this sort of powering the sorts of questions that we're answering? Thanos, I wonder, just from the first paper, I understand that was a really key part of the work that we did, was looking at 100,000 genome data.
Absolutely. So the first thing is that the participants have been sequenced with the same technology. So we can combine the cases that we've sequenced with this technology and be able to do the analysis. And also the fact is that we have also a lot of diverse data. So the analysis that you saw with different ancestries could only be possible because of the Genomics England participants, so that we can do in multiple ancestries an association study. Yeah.
Brilliant. Thank you. Thank you, Kenny. Thank you, Thanos, for joining us. A really enjoyable presentation, and I hope people get a feel for the power that the programme has had through that collaboration. We're now going to be joined for the cancer component of the programme by two of our members of staff from Genomics England, Nirupa and Alona. So, Kenny, thank you very much for joining us, and see you soon. So, welcome Nirupa Murugesu, who is our clinical lead for molecular oncology and an oncologist at St George's Hospital in the NHS, and Alona Sosinski, who is our scientific director for cancer genomics. This is going to be Nirupa giving us and Alona giving us a bit of a tour de force, an overview of a lot of the work that's been done looking at the 100,000 genomes cancer data set. So we're going to sit back and enjoy ourselves. We're going to hopefully have about 5 minutes for questions as well. But Nirupa and Alona, kick us off.
Thank you, Rich. So, thank you. So thank you for the introduction. So,
we'll start from the 100,000 Genome Project, and of course, patients were at the center of this program. And would you, Nirupa, take us through the journey of the cancer patient through our clinical environment?
Yeah, sure. So, just to sort of recap, as a cancer oncologist, patients would be identified for the project either within the MDT or in clinic. They would be consented, and the appropriate samples would be collected. Following that, the DNA would be extracted and then submitted for sequencing, and which would enable variant calling and automated interpretation through the Genomics England pipeline. And then this result was sent back to individual hospital trusts within the context of genomic medicine centres and reviewed by clinical scientists, and then these results were then looked at genomic tumour advisory boards with a minimum core rate of oncologists, clinical geneticists, pathologists. And I think within our own centre, we looked at somewhere in the region of 2,000 whole genome reports through the project. And then obviously that would then be passed on to the treating clinicians. So perhaps, Alona, you could run through a case and an example where this has impacted patient management.
Yeah, thank you, Nirupa. This was one of the patients that it was submitted, sample was submitted from the metastatic tumor from brain, and it was submitted as a carcinoma of a non-primary. So I will go quickly through what kind of analysis we run routinely on our automated bioinformatic pipeline, and what the conclusions we came up with for this patient. So we're looking at the somatic and germline variants for the patient and for this tumor, both small variants, structural variant aberrations, structural variants. Here is the short summary of the finding for potentially actionable variants for this patient. As you can see, patient tumor was carrying p53 mutation, and we also find a loss of function, frame shift fusions in driver genes, which are very typical for the lung cancers, RB1 and NF1. Then the next step of the analysis was to look at the tumor mutation burden. And of course, it's very important now with the introducing of the immunotherapies. So this is the classical way of looking at the tumor mutation burden. Different columns correspond to different tumor types, and overall this plot is showing about 10,000 tumors. On the left are the childhood tumors with very low mutation burden. On the right are the tumors with high mutation burden. So for this specific patient, we saw very high mutation burden, which is usually typical for colorectal lung melanoma patients. And the last piece of puzzle was looking at the mutation signatures, looking at the context of somatic mutations. And you can see that about 80% of the somatic mutations for this patient were CA transversion. We call it smoking signature. So when you put all this evidence together, it's very likely that the primary tumor for this patient came from lung. But of course, this is not the, so I went with Nirupa through the clinical part of the cycle, but of course it's not the end of the story. All the clinical information, all the genomic information for our patients go into the research environment, where it's available for our commercial partners, for our academic partners, GCIPs, and would you, Nirupa, take us through what is available now in the research environment?
Of course. So there were over 17,000 cancer participants, and the genomic data was linked to histological data so that the tumor type and subtypes could be gleaned. And you can see from this pie chart that there are nearly 3,000 breast cancer genomes, and then the next most common cancer types were next is colon cancer, separating out colon and rectal cancer, which is denoted as RIAD, and then sarcomas and renal cell cancers. So get an indication of the spread of the different tumor types. And I think importantly, they constituted tumor normal pairs using fresh frozen tumor samples established through pathways within the NHS with 100-fold coverage for tumor sequencing. And then alongside that, key clinical information that's required is stage, and so this indicates the spread of different stages that were collected from the different genomic medicine centres. And then on the right-hand side, you can see the breakdown again of stage by tumour types, where you see that there is variation, but there are certain tumour types such as melanoma and ovarian cancers where there are a higher proportion of stage 4 tumours. And then importantly, the genomic data needed to be linked with the clinical data. So from the curated Public Health England data sets, so the cancer registration data, the systemic anti-cancer treatments, so that all of the treatment regimens that the patients have received can be also linked alongside the genomic data, and then important information such as hospital episode statistics that gives an indication of where patients may have come back into hospital, where they may have been investigated for potential recurrence, and importantly, ONS data, so we can look for if survival and if this is also unfortunately related to their cancer. And so that's an overview of the clinical data, and because of this and through the foundation of that was laid down by the 100,000 Genomes project was the development of the Genomic Medicine Service, which we've heard about earlier on in the day, and this sort of highlights the existing tumour indications that are currently commissioned within the NHS. And as Sue mentioned earlier, the tumour types that will be available going forwards in cancer. And I just really wanted to highlight that the last category consists of metastatic cancers. So these are where patients have exhausted standard of care testing and treatment. And this is envisaged that this will be very much physician demand led, allowing for testing for patients that would be suitable for clinical trials. So moving on, we've sort of summarised the clinical information that's available, but alongside the reports, we know that there is a host of now cancer data analysis that's available. So if I ask you, Ilona, to perhaps update us on that.
Yeah, thank you, Nirupa. It was very hard to pick just a few slides from the wealth of the analysis that we run on our 100K cohort. But of course, all we do is very tightly link to the NHS priority, to the Genomic Medicine Services, and to the National Test Directory. And just to introduce you to the test directory, it was launched 3 years ago, but of course it's constantly updated. The most recent release was in October of this year, and it covers all the tests which are commissioned by NHS England for each patient in England, in this country. And it covers scope of the test, like which genes have to be covered, technology, and which patients are eligible for this kind of testing. And here we just summarized the data for the 100 cohort patients recurrence of the mutations in the genes, which are mentioned in the National Test Directory, mutations in the genes that make patient eligible for the targeted approved treatments that can also represent diagnostic prognostic markers. So I'm showing here only the top of the table. Of course, the list of the genes is much longer. They're on the rows. And columns are the different clinical indications. So for example, PIK3CA finally made its way to the test directory for the breast cancers. It's highlighted at the first column with the orange color. But if you go along the line, you can see that, for example, in the middle, patients with endometrial carcinoma, 50% of these patients have PIK3CA mutations. It doesn't, the PIK3CA inhibitor is still not approved for this clinical indication, but we envisage that with the expansion of clinical indications for this treatment, our test directory will also get expanded. So there is a lot of coming in the future here. I'm only showing actionability for the small variants, but of course you're not doing whole genomes for, reporting only, oops, it's a spoiler, only for the small variants. It looks like there is a lag. This plot is showing summary of actionability across different types of the variants. And you can see immediately justification, for example, why sarcoma, it's a third clinical indication here from the left, why it was included into the first wave of genomic medicine services for whole genome sequencing. You can see that more than 50% of sarcoma patients have actionable copy number changes or structural variants. And as Nirupa mentioned, gliomas are now also commissioned for the whole genome sequencing, so this GBM and LGG. You also can see that 80% of glioblastoma patients have actionable copy number aberrations, and whole genome sequencing is very suitable for accurately spotting this type of variants. What I'm not showing here yet is the pangenomic markers. And there is already MSI instability for colorectal cancers, homologous recombination deficiency for ovarian cancers in the test directory. And I just have a couple of slides covering what we're doing exactly now at the moment. It's just photosuppress for the homologous recombination deficiency. Why is it so important? Because it's already NICE approved that patients with HRD status are eligible for the treatment with PARP inhibitor. It's already in our test directory. At the moment, this testing is done by the Myriad panel in the United States. So we have to send material abroad. It's several weeks turnaround time. It's expensive, and we also have high failure rate. So why don't we do it from the whole genome sequencing? Algorithms for this were published already like 5-6 years ago. They just didn't make their way into the clinical environment, and this is what we're trying to achieve right now. We cannot run clinical trial. Very few of our patients are treated with PARP inhibitors because most of the patients were diagnosed in 2018, 2017. But most of these patients, more than 90% patients with high-grade ovarian carcinoma, were treated with platinum chemotherapy. And we can use it as a proxy of response to PARP inhibitor. So here is the, we put together genomic data with the data from national census on mortality, and we look at the survival of patients stratified by HR deficiency, HR proficiency. And as we expected, we can see that survival for patients with HR deficiency is much higher. In 3 years, it's 75% versus 50% on HR proficient patients. Okay, very good start. We can use, in this case, analysis was done by the cohort algorithm. And we already can see that we can use the HR deficiency based on the whole genomes as a prognostic marker for our high-grade ovarian patients. And this is the next bit of analysis we did for the HRD. We received a lot of inquiries also from our commercial partners of Can you do HR deficiency study for all 100K patients, put this data into the research environment? So following this validation of the algorithms, yes, we can do it. Here is the concordance between HRD status and BRCA deficiency, again, same set of patients. And we can see that all patients with pathogenic mutations, somatic pathogenic mutations, germline pathogenic mutation, and also deletions in the germline happily landed in the basket of HR deficient patients. There was only one exception on the left. And because we can put together genomic data with treatment data, with mortality data, with intervention, surgical intervention data, we can follow patients' clinical story, clinical journey, and unfortunately I don't have time to go through the details, but it's very obvious for this patient that came up discrepant. It's not likely that it was patient with HR deficiency because it passed away in a year after diagnosis. It responded to the therapy very, very poorly. So this is just to give you an example how genomic data can be put together with all the wealth of the clinical data in our research environment to come up with such validations. Thank you. Ready for questions?
Thank you both. I'm sure we should have just a few minutes for questions. That last point was really interesting about how you use genomic data alongside other sets of data that are available. Tell us about, perhaps starting with you, Nirupa, about some of the data that we're expecting to come in the future into the research environments.
Yeah, so I think, key data will be digital pathology and also radiology, and combining potentially genomics and radiomics. And I think Louise Jones later on in the afternoon will be discussing about multimodality platforms as well. So I think there's a real opportunity, and then there will be other omics such as circulating tumour DNA and opportunities to look at RNA sequencing as well. So there is a real opportunity to combine that alongside the clinical real-world data.
Great. Thank you. And in terms of sort of the types of variation that we see, Elena, you were highlighting in a number of those plots, the things that influence choice by the NHS in terms of what technologies they recommend for different variation types. We know that structural variation is really important in cancer in general. Can you talk us through about where you see the future in terms of the different technologies we, Genomics England, might be looking at, or NHS England might be looking at, thinking about structural variation and making sure we make the most of what we can interrogate in the genome?
Yes. We're doing a lot of comparison of the genomes with the standard of care, with the FISH data that genomic labs are doing in parallel. So we already saw that genomes coming up with a very high accuracy and sensitivity for calling fusions, for example, and other rearrangements. But I think it will be discussed later that we already start looking at the outcomes at the analysis of the long reads data, and how it can improve sensitivity of calling, how it can cover these black matters of the genome, for example, centromeric, telomeric regions.
Great, thank you. And I guess that's a really nice example, I think, of how through the 100,000 Genomes project, the sort of data that you've been talking about today has fed decisions by the NHS to inform their commissioning. And then we can work together, whether it's data that we, Genomics England, generate through our research programs we deliver in partnership with the NHS, or that there's data out there from other sources, help make those decisions about what the best bang for your buck is in terms of technologies, but also what data is really useful to have there available to researchers. And we heard Sue speak earlier about the desire of the NHS to bring panel data, for example, in to be available into the National Genomics Research Library for consented participants, so that it goes both ways. Perhaps just one final question bit of a sort of step back one for you, Nirupa, to close with is, tell us what you see as the experience as an oncologist in cancer genomics. Maybe if you sort of cast yourself into sort of 5 or 10 years, what are the what will have changed for you?
I think I anticipate that that cancers will be far more molecularly subtyped, and potentially we may move away from sort of more of a tumour agnostic subtyping or classification of tumours. And I think I would hope to see that we see a lot more molecular integration alongside our sort of traditional pathological diagnosis, and that, obviously then that impacts in terms of treatments and being far more precise in terms of precision oncology in how we manage our patients, and being able to predict, I think importantly, how they're going to respond to treatments.
Brilliant, thank you. That's a really nice note to close on. Just a reminder, we heard earlier, but in the innovation showcase section later this afternoon, we've got a couple of areas of focus on cancer, so please do join that section. Thank you, Nirupa and Ilona. That was a really enjoyable walk through some of the cancer data as we see it, and we're now going to thank you and welcome our next speaker. Thank you. So we're now joined online by Professor Matt Hurls, who is the head of human genetics at the Wellcome Trust Sanger Institute. Matt was hoping and quite excited about joining us in person here today, but he hasn't been able to join because he's unwell. So thank you, Matt, for joining us even online. We've been talking about the work that you are going to talk about today over the last sort of few months and in fact longer. We thought it'd be really nice to hear this story from you today, because I think it sort of pulls out some of the really nice features, of the great science that you and your team have been doing, but it's also stimulated discussions actually between ourselves about some of the opportunities it highlights in terms of working with data from our participants and the sorts of questions that we might be able to answer in the future. So what we'll do is start and hand over to you, Matt, and sort of talk us through what it is you found, and then we're going to have a bit of a discussion after that. And also for people online, please also think of questions that you want me to put to Matt as we go through.
Great. Well, thank you very much, Rich, and sorry I can't be there with you. I hope my voice lasts out for the next 20 minutes. So it's, as Rich mentioned, this is very much a kind of a vignette, which hopefully is illustrative of the kinds of things that can be done with the rare disease data in the Genomics England research environment. So the key to the success of this particular project has been the kind of teamwork between a set of analysts, data analysts at the Sanger Institute, the gel bioinformatics team with whom we've worked closely with, the clinicians of some of the patients that we've focused on, and also some functional collaborators in the US who are particular experts on one of the particular genes we focused on. So the thing that I'm going to be talking to you about is germline mutations, and essentially de novo mutations, that is new mutations that we have that our parents didn't have. We all have tens of these mutations. And it turns out they're a major cause of rare genetic diseases. And we and others have shown that through nationwide studies such as the DDD study or the PAGE study of a diverse set of developmental disorders, whether studied postnatally in the case of DDD or prenatally in the case of PAGE. And in both cases, a very large fraction of those diagnoses that can be made are these new mutations, these new de novo mutations, so where typically one of the two copies of a gene is damaged. And so it's important that we kind of then understand what is the causes of these mutations, and are there people out there who might be at higher risk of these disorders because of a higher rate of mutation. And what we know about the factors that influence germline mutation rate is that the predominant factor is how old the parents are at conception, and especially the father. So the older the father, the more mutations the child has. But everyone pretty much has 40 to 100 new mutations their parents didn't have. So we were interested to think, well, are there any outlier individuals, individuals who are hypermutated with an unusually large number of these mutations? And here we're just focusing on single nucleotide variants. And what we did was combine together data from the DDD study and from the 100,000 Genomes project. So almost 22,000 families where we've got DNA from the child and both parents that have been sequenced. And what we can see here on the left-hand side is the very consistent relationship in the gray dots between increasing father's age and increasing numbers of de novo mutations, and also with mother's age on the bottom plot. And there is a very characteristic mutation spectrum associated with these gray dots, which varies very little, certainly compared to somatic mutation processes, which is shown at the top here, this mutation spectrum. And one of the things we observed was that as you look at some of these hypermutated individuals, then what you can see is that they have very distinct mutational spectra that are different from the one that we see in the majority of individuals. And as the title says, these are pretty rare, these individuals that have these unusual. So there's only 12 out of these roughly 22,000. And hopefully you can see here that there are these 4 different examples of hypermutated individuals, which all have a very different spectrum of mutations from the ones that we see in most individuals. And hopefully you can appreciate that the middle of these plots and the bottom of these plots are actually quite similar to one another. So that suggested to us that maybe there's an underlying cause of this hypermutation that's in common between these 2 individuals. So one of the things that we did then was to take these hypermutated children and look at the mutation spectrum and try and see, can we identify mutation processes that generate these mutation spectra that we already know about through the work that's been done over the last 10 years or so on somatic mutation signatures. And just to orientate you, these different colors on the left-hand side are different mutational signatures that have previously been identified, and they've been given numbers. So what we know is that the mutational signatures seen in cancer that are mutational signature 1 and 5, that's the red and the blue, those combine together to create that normal mutational spectrum we see in the germline. And these other colors that you see are highly unusual. We don't normally see these in children in these de novo mutations. And what we could identify was this, that this gave us clues into what the mutation processes were that were aberrant in these children and indeed in their parents. And 2 of these children, the first and the third, with the largest number of mutations and the third largest number of mutations, turned out that all of these excess mutations were coming in on the sperm, and the father had a genetic DNA repair defect. And the first of these was a well-known DNA repair defect that's associated with a particular genetic condition called xeroderma pigmentosum. And using the healthcare data, we can see that the father really did have that disease. And that mutational signature corresponds exactly with the DNA repair pathway we expect to be perturbed. And then this third individual with the third largest number of mutations had a different mutation in a different DNA repair gene, one that hasn't previously been associated with a genetic disorder or indeed associated with cancer. And this is where our collaborators in Michigan did some functional work to show this particular mutation really does change the activity of this DNA repair enzyme. So there's a genetic cause of this hypermutation, which is pretty rare. But we also found that among these hypermutators, there was a very high proportion of parents, especially fathers, who had previously had cancer and had previously been treated with chemotherapies prior to conceiving their child. And what we could identify is this lilac color here, which was seen in common between those 2 individuals, which I highlighted earlier. That was actually a new mutational signature that hadn't previously been seen in cancer. And we managed to identify through working very closely with the health record data on the parents, but also with the clinicians who recruited those patients, going back and looking even deeper into the past at the parents' health record data. We could see that those 2 individuals with lilac colored mutational signatures had both been treated with a similar class of chemotherapy. One for Hodgkin's lymphoma, the other actually not for cancer, but for immune-mediated disease. And then 3 of these patients with this salmon pink color down here had a mutational signature which had previously been linked to platinum-based chemotherapeutic drugs. And all 3 of those fathers had cancers that are typically treated with platinum drugs and had had chemotherapy. So we think what we've identified here is that there are genetic and chemotherapeutic causes of hypermutation in these individuals, but they're very rare. Now, we also took home several lessons from this. So germline hypermutation is rare. In many respects, that's very good news because it means that common environmental exposures are not increasing to a very large degree the risk of these very severe genetic conditions caused by new mutations, so smoking, for example. We can also infer that actually the majority of germline hypermutated children won't actually have a genetic disorder. The risk on average is about 1 in 300 of having a new mutation that hits a really important developmental gene causing a disorder. And these hypermutated individuals only have up to 5 or 6-fold more mutations than we expect to see there, which would take the risk up from maybe 1 in 300 to 1 in 50 to 1 in 60. So still, a small minority of those hypermutated individuals are going to have a genetic disease. So we find that specific chemotherapy, certainly not all chemotherapy. So one of the things that we've been able to do is to use the health record data that's available on all the parents, go back and find all the parents that have had cancer and chemotherapy, and then look to see if their children appear to be hypermutated, and the vast majority don't, actually. So it's not all chemotherapies that can cause this. And so a couple of broad points that might be of more general interest is that this has only been possible because of the broad health data on all family members. And in addition to that, the ability to recontact the clinicians and get access through them to data that may be prior to digitization. So there's some heroic work done by one particular clinician here to go back through microfiche records to find out that that patient with nephrotic syndrome really had been treated with a chemotherapeutic agent. And that kind of passion and rigor in the clinicians that one can recontact is really important for distilling out these kind of key messages and key findings. So with that, I'm happy to take any questions or comments and also discuss more generic points with Rich and others. So thanks very much.
Thank you, Matt. So as you said on that last slide, it's a really nice vignette because it shows how in amongst 21,000 of the families who've joined our programs, you could find some really important but very hard to find messages, and then take advantage of what those families had agreed to, which is the ability to recontact the clinician for us to access longitudinal health record data. I know you've spent quite a lot of time thinking about the other sorts of questions that sort of approach might help you to engage with. Can you share some thoughts on where you've been thinking?
Well, certainly, one of the big challenges is of course, most of the rare disease patients still don't get a genetic diagnosis. And so discovery of novel genetic conditions is really one of the key aims of much of the rare disease research community. And the opportunity with the whole genome sequencing data is to look into the non-coding parts of DNA, whereas most other studies, including ones that we've run, have focused on the protein coding genes. So that's a really important motivating factor and something that we can really do with this data set, especially as it grows. I think another important thing is that this health record data with its longitudinal nature helps us to characterize the natural history of these disorders. And certainly when one starts thinking about how can we inform meaningful outcomes for clinical trials, we really need to get a good picture of that natural history. And so many of these disorders have only been identified in recent years, and we only have a very small snapshot of the phenotypic spectrum and how the disorder evolves. So it's really important that we characterize that, and the longitudinal nature of these data are very important in that regard. And then I think there are certain sets of questions where having the rich phenotypic data on parents is particularly informative and very unusual in the research domain. So it allows us to probe in much more detail the role of inherited genetic variation that might be and that might play. So not necessarily de novo mutations, but damaging genetic variants that a child has that the parent doesn't have, that the parent also has but doesn't have the disease or appears not to have the disease. Now, this gives us the opportunity potentially to identify individuals who are resilient to that damaging genetic variation, and further work on those resilient individuals may well reveal druggable opportunities for thinking about those diseases. So that's just a couple of ideas around the kind of unique nature of this data set and the kinds of analyses one can do with it.
Great. And a question first from Pierre Farmer. He's interested to know, can this sort of approach be used to monitor environmental effects, for example, on hypermutagenicity or genotoxicity, etc.?
So that's a really good question, actually. And I think there is a real opportunity as we think about how does this research resource evolve over time to think about how we link up to kind of drug-related toxicity and how we look, use the pharmacological data that's potentially available within health records, but also how we link up to the environmental data. So, what's the role of air pollution in this? Are there combinations of genetic variants and environmental pollutants that are contributing to some of these diseases? So I think there's an opportunity in using the very rich UK epidemiological expertise, especially in organizations like Health Data Research UK, to start to link genomic data with environmental data, with clinical data. And it's the full package that's going to be really informative.
Fantastic. And then a question from Joe Butler, which is about your take on where it is in terms of potentially other sorts of algorithms or other sorts of technology that will help us find the diagnoses that we're really struggling to find. Where is that? Is that through greater knowledge on existing data? Is it through new approaches looking at existing data, or is it new technologies? Where are the findings, those new diagnoses that we're missing, going to come from?
So it's a very good question. So we've done quite a bit of work largely within the DDD cohort to kind of look at what a bit the likely causes in the undiagnosed patients, and there's three kind of observations there. One is there is a contribution from non-coding genetic variation, but it's relatively small, at least in the way that we've looked at it thus far. The second is that there are still many de novo mutations that we've already detected in protein coding genes that are contributing, but we haven't yet got enough evidence to say that gene is definitely associated with that disease. And there's also a contribution that we've seen from damaging rare inherited genetic variants. And so what this really says to us is the most important thing we can do is aggregate the data together internationally. And so one of the things we've been doing is working with Genomics England to bring in the kind of biggest, largest exome data sets on similar kinds of patients so we can meta-analyze them in the genomics environment. And it's really a combination of scale of data, and that really requires international collaboration, and also looking at more complex genetic models. So not just these simple Mendelian models, but how might incomplete penetrance or variable expressivity of inherited variants operate. And that may well lead us down a path of certain genetic variants in combination with certain environmental triggers being one of the things that can give rise to a developmental disorder.
Great, thank you, Matt. I think that's a great note to finish this session on. I hope everyone has enjoyed getting a bit of a feel for the different areas of research that are happening at the moment, a bit of a feel for the sorts of data set and ability to sort of really work with our participants and with the NHS to answer some of the questions that we need to have the impact we want to together. To just highlight for people who are here with us in person and for those online, we're going to have a break now for 30 minutes and then please rejoin us for the innovation session. And that's a single track, so we're going back to a single track rather than having two sessions. But finally, to thank Thanos, to thank Kenny, to thank Nirupa, Alona, and to thank Matt, particularly from his state of sore throat and just about managing to join us online. Thank you very much for what's been a really enjoyable session.